Assalamu alaikum friends and welcome to my YouTube channel. I am Sikandar Kori. So let's start our topic.
Today in this video we are going to talk about codominance and multiple alleles. So let's start our video. First of all, what is codominance? It is a phenomenon of inheritance in which both the alleles of a contrasting character are dominant and expressed in heterozygous individual, neither masking the effect of one another. So this is the phenomena in which heterozygote expresses the phenotype of both homozygotes and there is no blending of characters. In human population, heterozygous sickle cell anemia, IA and IB alleles for the ABO blood group, heterozygous Tay-Sachs disease are the examples that show codominance. And now we are going to cross red bull with this type of genotype with the white cow having genotype R2R2. And during the gamete formation, each gamete will receive single copy for the gene. And the progeny of this cross will have roan coat color. So we call it as roan cattles with the genotype R1 and R2. Roan basically is a mixture of red and white hairs, and we see the patches of red hairs and white hairs over the coat color of these progeny. So in this genotype, these both alleles are expressed specifying their respective protein or pigment. And there is no intermediate phenotype, and this heterozygote have the phenotype of both homozygotes of both parents. And if we cross these roan cattles with this type of genotype, suppose we have gametes for these individuals, and the union of these gametes will have this progeny, where we have one individual with a red coat color, two cattles with roan coat color, and one cattle having white coat color. And now we are going to talk about multiple alleles. In our previous videos, we have talked about that a single gene have two alternative forms or alleles, but this is not the case in multiple alleles, where the single gene for a trait has three or more alleles, such as human leukocyte antigen genes, which specifies the protein which is part of the major histocompatibility MHC complex in humans, such as HLA-A gene may have about 60 alleles, HLA-B gene have 110 alleles, HLA-C gene have about 40 alleles. Another well-known example of multiple alleles is ABO blood group gene, where a single gene have 3 alleles. So these kind of gene are called multi-allelic gene and trait called multi-allelic trait. And now we have to discuss ABO blood group system in more details. So the gene for ABO blood group have 3 alleles such as IA, IB and small I. And here I stands for isoagglutinogen. Allele IA specifies the enzyme which add galactosamine to the surface of RBCs. Allele IB specifies the enzyme which add galactose to the surface of RBCs, whereas small I specifies the enzyme which add no substance to the surface of RBCs. So these substances which are expressed on the RBCs can initiate the production of antibodies by the B cells and so called antigens. And we see the clumping of RBCs when a mismatched blood type is transfused to the person. So the alleles IA and allele IB are codominant with each other, and both are dominant over small I allele. As we all know that every individual inherits two copies for a gene, and so there are 6 possible combinations for these 3 alleles, such as this combination of alleles for the individual have A blood type and will have A antigen to the surface of RBCs, and this individual can receive blood from the individual having blood type A and O and can donate his blood to the A and AB persons. And these combination of alleles, the person have B blood type and will have B antigen to the surface of RBCs, and this individual can receive blood from the individuals having B and O blood types and can donate his blood to the individuals having blood type B and AB. And this combination of alleles IA and IB will have a blood type AB, and they have both of these antigen to the surface of RBCs. This is the universal receiver, can receive blood from any type of the blood and can donate blood to only the person having blood type AB. Individual with this type of genotype have the blood type O and will have no antigen expressed to the surface of RBCs. This individual can receive blood from O blood type, and this can donate the blood to any individual with the blood type. So this is all about our today's topic.
Thank you for watching our video, and please like, share and subscribe to our channel.